So we spent a lot of time talking about different methods for association with binary traits, with quantitative traits, and doing GWAS, as well as how to do association with gene-based pathway analysis. But one thing that you often have to be concerned about in genetic association study is that of confounding due to population structure. So has everyone heard of population structure before? Okay. So it's a pretty common term, particularly in the field of genetics, where you're concerned about false positives or spurious association due to differences in individuals' ancestry, which may be related to the trait of interest. Okay. So it's pretty well accepted that humans spread around the world many thousand years ago out of Africa, and due to migration and genetic drift, that led to the large-scale diversity across the genome among different populations. Okay, so so if you look at the genome of individuals across populations, you'll see that certain populations are more closely related, for example, the European population, but you have other populations that are highly divergent. For example, Africans and those from East Asia, they have very divergent genomes. And so when you're doing genetic association studies, you're often concerned about individuals in your sample who may have different ancestry, and that ancestry may be driving the association, and it may not be, then the association you're seeing, it may not be due to the true causal variant or markers that are tagging the true causal variant. So inference on genetic ancestry is critical for a variety of areas in genetics or population structure. You often see it in the literature. These are just genetic ancestry differences among individuals. Okay, so there are certain individuals that cluster. Right? If you look at their genome, they have more similar genetic data than others. So you see some clustering of the individuals in your data. So that means population structure. So certain individuals seem to be more closely related genome-wide than other individuals. And so identifying or inferring genetic ancestry differences is essential for a variety of applications, including population genetics, genetic association studies, personalized medicine, forensics, and linkage analysis. And so with the advancements in high throughput genotyping and sequencing technology, they have facilitated the investigation of genetic differences among individuals at remarkably high levels. Okay? And so there have been a variety of methods that have been proposed for identifying genetic ancestry differences using high-density genome screen data. Okay, so PCA, performance analysis, is by far the most widely used approach for identifying and adjusting for ancestry differences among sample individuals in genome-wide association studies. Okay, so PCA can be applied to genotype data, and where you calculate principal components that explain differences among sample individuals in the data. And so the idea is the top principal components along a certain axes of variation is supposed to reflect genetic variation or genetic differences in ancestry along that axis, along that principal component that's reflecting ancestry along that component. Okay, so individuals with similar values for a particular top principal component are expected to have similar ancestry for that axis. Okay, so the top axis is supposed to reflect the most variation, and then the second to top is going to be the second most variation. Okay, so the idea is if you do PCA on genotyping data, differences among individuals should be captured by the PCA, and the idea is that genetic difference should tell us about ancestry of individuals. Okay. So how is PCA performed using genetic data? So suppose we have genome screen data on individuals in our sample, and so we'll assume that we have genotype data at S SNPs, and so S typically is going to be very large. So in our genetic association study for transferrin, S was 250,000. So S can be very large, even a million, for example. So for SNP S, we're going to define, so for SNP, we're going to define the genotype data to be 0, 1 half, or 1, or it could be 0, 1, or 2. I mean, so it's just additive model where if I'm coded as 0, 1 half, or 1, just the additive model, you want to copy reference those divided by my that too. But whether or not you do 0, 1/2, or 1, or 0, 1, or 2, it actually does not matter because we're divided by a constant, which is 2. So the idea is you define a matrix, okay, that's essentially for each individual at each marker, you standardize this individual's genotypic value. So you subtract the mean value, which is going to be the allele frequency, and you divide it by the square root of p times 1 minus p. So this Z matrix essentially is a standardized genotype score for individual I at SNP S. Okay, so for each individual, we center, okay, we center it, and then we standardize it. Okay. And then what we do is we essentially look at all possible pairs and look at the genetic similarity. So you can obtain a matrix by multiplying these standardized genotypic values. So for each individual I and each individual J, we have a measure of how similar they are. So we measure, for example, if you look at the introduced matrix, we have individual I's genotype minus the average of the frequency, individual J's genotype minus the average divided by p equals 1 minus p. So this is essentially a measure of the average correlation of individuals I and individuals J's genotype. So individuals who have similar ancestry, okay, are going to have positively correlated values, right? Because if GI, for example, if individual I has similar ancestry and if they're from the same population as J, if this value is high, for example, let's say this person is more likely to have a 1 here and a 1 here. So if this is high, then this might look likely to be high. So this is going to be positive. If this is going to be 0, this is negative, this is negative. Negative times negative is positive. Okay? If I and J have different ancestry, then you're going to have a lot of situations where, for example, this is going to be positive and this is going to be negative. So that's going to have a negative correlation where one is going to have a high value at a marker and the other one's going to have a low. So this gives us a measure of how similar individuals or different individuals are. People who are from similar ancestry backgrounds are going to have correlated values. So this is an average correlation standardized for individual I and individual J's genotype. So it's an average correlation of an individual's genotype across the genome, and you do this for all pairs of individuals. Okay, so that's the genetic relationship matrix. Okay, some people call it the genetic similarity matrix. Okay, so we're just measuring the correlation for all possible pairs in your sample. Question? So that's the question. So it's recommended to do some type of pruning for LD because a lot of times these SNP arrays, for example, there are certain regions that are overrepresented, for example, gene-rich regions. And so you really want a good representation of across the genome. So you probably would do some type of LD pruning. From my experience, I have found that it really doesn't matter that much. I can get similar results, and you'll see in your example data set that you're going to be playing with after the lecture, that you kind of get similar results even if you do LD prune or not, because the genome is so long, and so there's enough independent blocks that even though you do have correlated data, correlated SNPs in certain regions, there's enough independent blocks that you still capture the appropriate structure. Right. But yes, so you can do LD pruning, and typically that is what is recommended. Now there are something you need to be concerned about with PCA. There are certain regions of the genome that can hijack your PCA, for example, like on the HLA region on chromosome 6. So there's certain region inversions where they suggest that you remove those because they will completely hijack your PCA. Okay, so but in general, you probably will do some type of LD pruning. Okay. So the idea is once you have this genetic relation matrix, genetic correlation matrix, okay, then you just do principal components analysis on this genetic relation matrix. So this is our matrix, you just do PCA. So what is PCA? It's just single value decomposition, okay? So the orthogonal axis variation that best explain the genotypic variability amongst the in-sample vendors are identified. So you just do an eigen decomposition or single value decomposition of this matrix. So every square matrix can be written in this form. V transpose, where V is an n by n matrix with orthogonal column vector. So V are the essentially principal components. And then diagonal matrix, so this D is a matrix that has zeros on the off diagonals and lambda 1, lambda 2, all the way up to lambda n on the diagonal. And so you can decompose any square matrix into eigenvectors, the eigenvalues times the eigenvectors transposed. So the eigenvalues are in decreasing order. So the first principal component that corresponds to the largest eigenvector is the top PC, the top principal component. Okay. The eigenvector that corresponds to the second largest eigenvalue is the second most important principal component, and so forth. Okay, but the top principal component is the one that corresponds to the highest eigenvalue, and it explains most of the variation in the data. Question?
Yeah, so the terminology is just a little bit different than like traditional PCA, which is continuous structures, right? So I guess I'm wondering, are these being treated as discrete characters then?
So when you say, so PCA can be used on any continuous and discrete, and
well,
that's
the
analysis.
BCD, matrices is looking at variance.
Yeah. Okay. So note that SNP genotype data is discrete, 0, 1, or 2. This is no longer discrete. This measures the correlation, right? So even though you have discrete data, right, we're standardizing, subtracting the mean, and averaging. It's no longer discrete. So if you look at this matrix, it's correlation, right? So it's not. So you're not doing a PCA on a discrete matrix. I mean, this is a measure of how correlated two random variables are. And so whether or not the random variables are discrete or continuous, the correlation is going to be continuous because it's the measure of some similarity in those random variables.
Is a principal component different from a line of regression, or is it just a generalization?
So with regression, okay, so with regression, we're typically talking about there's some outcome of interest, right? And we're trying to kind of like predict, outcome from some predictors, for example, right? Here, we're not doing any type of outcome prediction. This is just genotype data. There's variability. Can we determine which component is explaining these variability, these differences among individuals?
Okay.
So there's a little difference. Typically with regression, there's an outcome we're trying to predict.
Okay.
Yeah. But you will do, once you have the principal components, for example, you might want to do, if you, like I said, you might want to see how well your principal components corresponds to known ancestry. And then people will do a regression model using the top principal components to see if you can predict ancestry from your top principal components.
Okay.
Question?
So would you hope to have as little principal components as possible? So would that mean that the population that you're studying is not very diverse and they're like from similar background? And then the more principal components you get, then the more diverse your population is.
Yeah, okay, so that's important. So when we talk about principal components, so we can talk about subtle structure or diverse structure. So I'll talk about like even subtle structure within like Europeans that you really can't even tell, where they are. That can have a huge impact, even if it's quite subtle. Yeah. So it's going to depend. If you have large differences among individuals, you probably want to do a separate analysis, right? So if I'm doing analysis and I have, because somebody says I have like data on like African Americans and some Asians and Europeans, I don't want to analyze them all because it's going to be very difficult to kind of control for and confound it. You're better off doing it within the Europeans, but even within the Europeans, there's a lot of differences among individuals. So, for example, we get a lot of data, and everyone looks the same, but then we find some like clustering. There are Ashkenazi Jews, and so there's, and some who are from like Finland or Northern Europe. I mean, so that cluster, can impact even if you think your sample is somewhat homogeneous. And I'll talk about that a little bit more as far as like differences among individuals, groups that you think are quite similar. So, principal components analysis of Europeans. So the top principal components are viewed as axis variation that reflect genetic variation due to ancestry in the sample. So individuals with similar ancestry should have similar principal components along a particular axis. Okay, so John November published a nice paper showing that in European samples that the top principal components corresponded quite well to the map of Europe. Okay, so in this study, what they did is they took samples from all these different countries in Europe, and they only included individuals who have both pairs of grandparents, all of grandparents originated from that same country. Okay, did a genetic relatedness major, genetic similarity, look at all possible pairs, did PCA, and this is what they obtained. So you might have seen this figure before. Okay, so this is the map of Europe, and you see that top components corresponded quite well, okay, with geography. Okay, so this is within Europe. So you see, for example, Portuguese and they're clustering here. And you see, all these different Italy and so forth. Like there's a nice correspondence with PCA in the map of Europe. Questions?
What was like the sampling?
Yeah, so I think, like I said, so I'm going to refer you to the paper. From my understanding is they went to each of these different, so they had data, for example, in Portugal, and I don't know what the sample was, I don't know what they were studying, but then they only included individuals who had both grandparents that were from Portugal, right? And so, and same with Spain and so forth. So only individuals who had both grandparents in that country. So, but I will refer you to the paper. That question.
How do you decide how many pieces to include in your model?
Yeah, so that's a hard question. I mean, to be honest with you, that's a hard question. So there are a lot of ways to do it. If you're doing genetic association studies, one thing you often would do, you'll do PCA plots. So at some point, there shouldn't be any structure. If you're seeing some pattern in the data, there's likely that component is separated individually based on ancestry. If you get to a PC where it's just a bunch of just noise, where there's no obvious pattern, then that's way. So that's the one way of diagnostic. There are other ways to do it too, is to look at the eigenvalue. So I mentioned that the top principal component is the component that corresponds to lambda 1, which is the one eigenvalue that's the highest. So some people will look at what they call sweet plots, where they plot the lambdas, and once the lambdas become stable, then that's their cutoff point that they use to say, okay, you know what? There's no difference from lambda 4 on, and so likely there's nothing really going on after that point. But there's no real answer to that. But people use different diagnostic tools for that, and so you can actually calculate the proportion variance that's explained by an eigenvector using the lambda. So for example, if you want to know the proportion variance explained by the top principal component, lambda 1, okay, is lambda 1 over all the lambdas, lambda n. And so this is the proportion variance explained by that principal rho variance explained by, for example, PC. So that's kind of like the formula. So you often see in papers where they'll report that. That's all they're doing. It's just an eigenvalue by some of all the other eigenvalues. So within Europe, you see a lot of variation. Okay, so the question is, what would you expect to see within a single country of Europe? And so there was a paper published a few years ago where they did principal pronoun analysis in a Finland sample, which seems to be, we expect to be quite homogeneous, right? So this is what they got. Even within Finland, okay, they found that there was population structure. So even in samples that you think are relatively homogeneous, okay, there could be structure, right? And so, question?
Is it not a trait that you're using to give that
This has
whether or not there's structure?
no structure exists no matter what, right? We didn't do any this is genotype data, right? You're going to be concerned if you have a trait that's associated with ancestry, with these differences. So, for example, I don't know Finland, but I mean, an extreme example would be, let's say, for example, these individuals are more likely to suffer from asthma because this region here is by a major highway, right? And you do a genome-wide association study for asthma. Okay, what's going to happen? These folks are more likely to have asthma because of environmental factors, right? Any SNPs that are showing differences between this group and the other group, you're going to get a signal, you're going to get an association. Because there is an association, but it's not a direct association through genetic data. It just is just telling us, okay, these folks are different, okay, than these other folks for this particular SNP. And on top of that, these folks are more likely to have the disease of interest. So you do have an association, but it's a false positive. Okay? So we're going to talk about issues with population structure and genetic association studies, but this is just to let you know that even if you think your sample is homogeneous, depending on your phenotype, you can run into major problems. So even with traits that aren't even genetic, they start looking genetic because of this association. And this is within Finland. So you wouldn't be able to tell the difference from them just looking at them, right? This is just genetic data. This is fine scale structure. Now, if you start talking about rare variants, it's going to be even more of a problem because rare variants are often population specific. Okay? So common variants you see in all European population. Rare variants might cluster in a certain geographic region where you only see this variant. That's what they found in the 1,000 genomes. Common variants, you observe these common variants in all of the different European populations. Rare variants were often specific to a particular region in a particular country. So you're even more concerned about issues with population stratification when you're talking about rare variants. Now, it's an active area of research right now, how to infer population structure with rare variants versus common variants. A wrong thing that people have been doing is for these rare variant association tests, and I'm putting in the card for the horse a little bit, because I think Mike's going to talk about the rare variant association test, is they do PCA based on the common variants, and then use the principal components for the common variants and adjust for the PCA for the common variants in the rare variant association test. But association testing with rare variants is complex because population structure inference with rare variants can be quite different than common variants. As I said, actually, there's more clustering with rare variants, right? Because certain rare variants only occur in certain groups. So that's something just to keep in mind. Okay. So what about admixed populations? So there have been a number of recent ongoing genetic studies that have focused on admixed populations. So these are populations that have been derived within the past few hundred years that have ancestry from two or more populations from different continents. Okay, so admixture occurs in the past because of a variety of historical events, like the transatlantic slave trade, African Americans, right? So brought the slaves from West Africa over here to Europe and Brazil and South America and so forth. So there was a lot of admixing there with the Europeans and the African slaves. Okay? Colonization of the Americas. Okay? So all the Hispanics, Latinos, South Americans are all admixed. All have European ancestry, Native American ancestry, some African ancestry, particularly those regions that actually where slaves from Africa were brought to those regions. So their genomes are even more complex than the typical European. So if you look at the genome of an admixed individual, so at some point these were populations that were isolated, came together due to historical reasons, slave trade, colonization, and then the first generation looked like this. The blue color corresponds to European genome, for example. The red could be Native American genome. Okay, so colonization occurred. Okay, South America, and the first generation. You see that the first generation, you see they came together, and then the current generation, the first generation, for example, is 50-50 for those who are admixed. And then if you look at the current genome of an admixed individual, you'll see that it represents a mosaic of blocks, where you see blocks of European, blocks of African, blocks of European. So it's actually a mosaic. Why do we see these different blocks? Any thoughts on that? Recombination. Okay? So there's been a lot of work done where they're trying to, based upon the length of the blocks, the number of the blocks, they're trying to determine how many generations since admixture, right? So you can kind of infer that based upon how many blocks there are, because every time you see a changeover from African to European to African, you know a recombination occurs between chromosomes. And so there's been some active research done on, can you infer how many generations since the admixture event when these two separated genomes came together and started mixing based upon the current generation, and looking at how many switches that admixed individual has on the chromosome. So one thing I should point out is that most of the things we've been focusing on have been with the autosomes. Okay? The X chromosome, Y chromosome is a different story. So for example, if we think, let's think of the Latin Americans, Hispanics, for example. What would you expect the Y chromosome to largely be derived from, and what would you expect the X? So the X is going to be largely native, right? Spain wasn't sending their women over there to colonize, right? They were sending the men. Okay? So if you look at the X chromosome, X chromosome has a lot more Native American ancestry than the rest of the genome. Okay? So if you look at a genome, for example, of a Mexican, it's almost like for autosomes, it's almost 50-50 for Mexicans. And I don't want to group Hispanics together because Hispanics are just so complex. So we're involved in this Hispanic Community Health Study that we're running here at UW. They're very complex. I mean, it's very different genetic profiles for Hispanics who are from the Caribbean, Puerto Ricans, Dominicans, Cubans, versus Mexicans, South Americans, Central Americans. Okay, very different. So, but one thing that is consistent is that there's higher native ancestry on the X than the autosomes because of this phenomenon. So there's information there about history, about what occurred prior to, and you can see that in the current generation. Okay. So there could be a lot of genetic heterogeneity within an admixed population. So they have admixed populations have largely been excluded from genetic association studies. I mean, recently now the NIH has put out an initiative to start doing studies in more diverse populations. But, I mean, there's a lot of complexities here because of the differences among individuals at different parts of the genome. And on top of that, you see a large heterogeneity. So, for example, when we're looking at, for example, Mexicans, for example, I mean, you have some Mexicans who look almost 100% European. You got some Mexicans who look almost 100% Native American, right? So you see a wide range of ancestry differences among individuals. How do you account for that in the analysis? So the statistical methods, haven't been developed well. Okay, they're at the infancy of how to even analyze admixed genomes, okay, which is some of the work I've been focused on recently. Okay, but how do we determine these blocks, for example, or what portion of the genome comes from European ancestry, African ancestry, right? So there's some methods out there to infer ancestry in supervised learning methods. So most methods use like a hidden Markov model or expectation maximization algorithm to infer ancestry. So if you don't know what that is, don't worry about it. The idea basically is that we don't observe where these points of the genome come from. We don't observe that this block comes from Europe, this block comes from Africa. We don't see those blocks. What are the data that we receive? So what's the data? We receive genotype data, sequences or SNP data that may be unordered. Okay. So this is an admixed individual. We want to know, for example, what proportion of his genome comes from these different ancestral populations. Okay. So these methods that are supervised methods for inferring genetic ancestry essentially use reference population. So there's a lot of data out there freely available. HapMap, okay, everyone's heard of HapMap. So HapMap data is available. HGDP, 1,000 genomes, all this is freely available data. So what they do is they take the samples that are from Europe, samples that are from West Africa, and samples from Native American. But Native American samples, there are no really good Native American samples because Native American samples are all admixed, right? There are no pure black Native Americans anymore. So even that is somewhat problematic. But in any case, let's say they were. The idea is you look at the stretch of the genome, and if you see stretches that matches a particular group, then that gives you information that this whole stretch comes from European or African. So essentially, it takes blocks of DNA from an admixed individual and it tries to match it up with the reference population samples. And based upon that, you can get an estimate of what proportion of the genome comes from these different groups. All right? Question?
So I wonder, you can use this to study where people came from, but is it a tool you can use to track a specific disease phenotype? Is it a good tool or bad tool to try to do that?
So you mean where a disease came from? I guess it's hard for me to visualize how that would play out.
Well, I'm just trying to sort out, would the only advantage be trying to figure out where somebody's come from, or could you use knowing where the segments came from to try to isolate a disease-causing mutation?
Yes. Yes. The answer is yes. So there's work and methods for admixture mapping. So for example, if you have a genetic variant, so let's say we have these local ancestry estimates. So let's say, for example, let's say we have local ancestry. So there are methods that do global estimates, which is average a genome. But lately there's been methods that actually can quite accurately estimate at a particular location. What do you have does this allele come from Africa or European as an allele, right? Because the thing about this is that these chunks are many megabases long. So we're not talking about like short sequences. We're talking about many megabases. So when you have a chunk of ancestry, a block of ancestry, we're talking about many million base pairs long, that whole chunk, because these are recently admixed within the past few hundred years. So there hasn't been that many recombination events. So admixture mapping, which a lot of people have been doing, those who work in that population, is it tries to say, okay, at this location, are individuals who have the disease more likely to have an allele that came from Africa, an African-derived allele. And so that's the idea. So instead of SNPs, where we count 0, 1, and 2 copies of the reference allele, we model, can we predict outcome based upon whether or not you have 0, 1, 2 copies of the reference allele? We ask, at this location, can we predict outcome based upon having 0, 1, 2, or 2 copy of an African-derived allele? And if you're more likely to have the outcome or have increased phenotype value for those who have African allele at this particular marker, then that kind of gives you some idea that the variant that is causing this trait or influencing the trait is in this region. So there is some work done on admixture mapping, and so there's a lot of literature on that. Okay. Question?
Do you have like a name of a recommended program that looks at this?
Yeah, tons of programs. So Admixed is the latest one out of Carlos Bustamante's lab out of Stanford. So Admixed, American Journal of Human Genetics, it was published last year. That's what we've been using. Our collaborator, Hua Tang at Stanford, she had her own program, Saber, but Admixed seems to do the best. Okay, so that's for estimating local ancestry. For global ancestry admixture, so John Novembre's program, he also did the PCA with the European. He has that software actually works quite well. It's called the Admixture software. But if you're trying to get local ancestry, so admixture just gives you average genome-wide ancestry. To get local ancestry, then you use Admixed. Saber is another one. Yeah, just do admixture mapping and you'll find it, yeah. And then if you're doing admixture mapping, then it's similar to GWAS, except instead of 0, 1, 2 copies of a reference allele, it's 0, 1, 2 copy of an ancestral allele. So it's similar association, linear regression, logistic regression, it's the same. And so there are pros and cons with admixture mapping. The con is that you're not going to identify anything. If, for example, let's say you have a variant that's causal. So there's this variant, you know it's causing, for example, high blood pressure. And this variant you see in equal proportions in the Europeans and the Africans, you have no chance of finding it with admixture mapping, right? Because that marker that's causal, you can have it if you have European ancestry or African ancestry. It's only going to, you're only going to get a signal if you see large and low frequency differences of the causal variant in the ancestral populations. Okay? So that's one thing to do. But on the other hand, because the blocks are so long, okay, you don't have so much of an issue with the LD with SNPs. Because the LD with SNPs aren't that long. So you can target, if you do have this, you can get signals where you can't with association studies, right? So for association studies, what do you need? You need to make certain that you have the variant and you have a marker that's tagging it well. For here, you don't need tagging because this whole region is tagging. Okay? And so some have argued that admixture mapping can help us with rare variants as well, right? Because this is tagging both common and rare variants. This just says this is your ancestry. So if the rare variant comes from an ancestral population, you should be able to pick it up. Well, you can't pick it up with single SNP or common variants, right? Because there's not strong LD with common and rare variants, but you should be able to pick that up with admixture mapping. So this is still at its in infancy, to be honest with you. I mean, like, these are open problems. How to do this. I have a PhD student working on this problem right now, how to do association mapping using mixed models and things of that nature. So if you want a dissertation topic, this is it. It's wide open. Yeah, and there's been a big push to try to analyze admixed genomes. So anyways, there's tons of methods out here. HapMap contains genotype data on admixed individuals. The Mexican Americans are also admixed. And the ASW admixed. So in your lab for today, you're going to be actually analyzing this using PCA. So I have gave you data from Mexican Americans from Los Angeles, the African Americans from Southwest, and these are data from HapMap. Okay. So just to know, I did Admixture. So this is the software if you want genome-wide average ancestry. I applied it to this data set where I try to estimate the proportion of ancestry for African Americans, the ASW, African American Southwest Texas, and the Mexicans in Los Angeles. And I used 3 reference population samples. So I used the HapMap Yoruban from West Africa to represent African ancestry. I used the HapMap CEU from Northern and Western European as European ancestry, and the HDP Native American samples. Like I said, they're not really pure Native Americans. These are just samples from the Americas, since we've actually had mixed. But it works quite well, even though they're not pure Native American, as the reference population for Native American ancestry. And this is kind of what I got. So in this plot, these are the ASW, this is the MXL. Blue represents European ancestry, red is African ancestry. So each individual is represented by a vertical bar. So you see here, for example, this individual in ASW almost has 100% African ancestry. So the genome is almost 100% African derived, but a little bit of European and a small amount of Native American ancestry. So as you go along, you see some individuals who have more European ancestry. These are the Mexicans. Right? So you have some Mexicans in the MXL who have almost 100% Native American ancestry. I think this one's almost 90%. Where you got other individuals who are almost 100% European ancestry. So even within the admixed population, you see a broad range of ancestry among these individuals. So here are the estimated ancestry proportions. Okay, so around 49% European and 44% Native American for Mexicans. Small African ancestry in the Mexicans, but not negligible. And for the ASW, that's about what you expect, about 78% African and about 20% European and very little Native American ancestry. Okay. So with that, you're going to do an exercise where you're going to do PCA using this data that I just presented for the HapMap. So you're going to infer essentially or infer answer differences using principal components analysis. Make sure you're doing session 5. Session 6 is on the back. Make sure you're doing that session. doing session 5, session 6 is on the back.
So I was creating a PCA using Supervised, and then one of my commentors was like, no, you should use Admixture because it like does something different.
Down to the same it's different days, all of it. It's just connects generation matrix. I mean, they may be minor tweaks as far as how you calculate those frequencies, but they're all All right. All right, let's do the tutorial. So you want to load the libraries and then set your working directory. Okay. So count the number of individuals that here. Dimension of fam. So the 604 individuals. Okay. So how many SNPs are available? Okay, so there are 150,872 SNPs on the individuals. So you want to read information that has information on which individual. Sorry, let me set the working directory. All right. So this is information about the population. We can use the table command. So we have 203 Yorubas, these are the West Africans, 63 Native Americans, 86 Mexicans, 165 Europeans, and 87 African Americans from Southwest Texas. Okay. So if you wanted a tutorial of SNP relate in GWAS format functions and packages, then that's the website you can go to for that. That's what I did to understand what's going on. So you want to create a file. So the SNP relate package, so just to let you know, so Plink is a very useful program. Like a lot of programs that are developed now are developed so that it can take Plink files. Okay, so Mike is going to cover some of the different ways where you can create a VCF file, for example, you don't sequence data so that you can input it in Plink and do the data manipulation very fast. So this is similar. This allows you to have a Plink file. So these are Plink files. Read it into R, and it creates a GDS file that can easily be used in R. Okay, so it takes a Plink file and then reads it into R. So here I'm converting the Plink files into GDS files, and I'm naming it IPC genome. Okay. So I'm going to call it my genome file. So what does it look like? So for example, if you want more information about it, so this is just, sorry. So this is just like, who are the sample IDs. So essentially it takes the genome file and then you tell it to give me the IDs of the individuals. Okay. You want it to look at the first few SNPs. Okay. And these are the first few SNPs. So if you wanted to extract SNPs or genotype data, you can easily do it in this with the SNP relate package. Okay. So if we wanted to, for example, look at a histogram of a particular genotype data, if you want to extract genotype data for a SNP of interest, let's say you're interested in this particular SNP, then you can extract it using the SNP GDS kit genome, and it gives you the genotype data for the individuals. For example, G, 0, 1s, or 2s. Okay. So if you wanted to perform a principal components analysis, okay, you would just do SNP GDS PCA. So it takes the genotype file. Okay. It's going to create a genetic relatedness matrix, do PCs, PCA on the genetic relation matrix, and then it's going to return eigenvectors and eigenvalues. Okay. So that's what I'm going to do right now. So it's doing all this under the hood, but what it's doing is exactly what I presented in the class. It's creating a genetic relationship matrix for all possible pairs, or genetic correlation matrix. Then it's doing a singular value decomposition and giving it the top principal components. All right. So it's completed. Okay. So it's letting you know that 604 samples, 2,872 SNPs, and some of the ways that it tries to calculate things pretty fast. Okay. So here I'm going to create a table of just the first two eigenvectors and eigenvalues. So what does this look like? So here, so these are just the top two principal components, top two eigenvectors. So each individual has a coordinate along each of the principal components for eigenvectors. So sometimes you hear eigenvectors or principal components, they're the same thing. The top eigenvector is the first eigenvector is the top principal component. Okay, so individuals who have similar values along this eigenvector are going to have similar ancestry along this axis that's representing this ancestry. So let's plot eigenvector 1 versus eigenvector 2. This is what you get. So you do see some structure. Be nice to have some color on this figure so we can know who is who. So for this, we can incorporate population membership. Okay. Okay. So now here's a plot. Okay. Where we have the individuals color coded by their ethnicity. Okay. Question?
I've been told to use Eigenstrat to calculate this. Does this compare? Or
Same. They're just doing single value decomposition on the genetic relation. Yeah. So it's the exact same. Yeah.
From this plot, would you be able to tell which samples are outliers, or would that matter?
Yeah, so here, I mean, nobody looks like an outlier, right? I mean,
Right.
there's
But
no.
if you had like one cluster and then you had one individual that was like way away, would you consider removing that individual from your?
Yeah, I mean, yeah, possibly, right? But everything's relative, right? So how you define an outlier? And why is it an outlier? A lot of times you want to investigate why is this person outlier? Is it a bad sample? But yeah, so if you think this outlier point is going to be driving or have high leverage on your results, then yeah, you might want to remove that person. Okay. So here, for example, in the African Americans, so what can we say about the African Americans? All right, so you see that the African Americans are largely African, you see closer to. So what is, for example, what is the x-axis? What is the y-axis telling us? How is it separating individuals based upon what? Interpret this plot. What is y-axis? So the top principal component is separating individuals based upon what?
A certain marker.
Not marker.
Genotypes.
What ancestry? African, non-African. Right? So first PC, these here all have a lot of African ancestry. These down here do not. What is the second PC separating? Based on what? European and Native American. Okay? So the first PC is dividing the data basically into two groups. Those that have a lot of African ancestry and not a lot of African ancestry. PC2 is separating based upon European versus Native American. And you see the admixed individuals don't really cluster. You got some admixed folks who look like 100% European. You got some who look almost like 100% Native American. Okay? And so that's because of the admixture. They have ancestry from multiple groups. Those who are here, for example, I'd imagine like one of these people here, for example, so this person has like European ancestry, but if they're closer to this way, they're actually too. If they're in this person here, what would you probably say about this individual? This person probably has one of the highest Native American ancestry as well. Okay. So there's information there as far as ancestry goes. So if you wanted to do a scatter plot of the top 4 PCs, we can do that as well. Okay. And remember, I told you you can plot the proportion variance explained using this formula here. So the top PC explains 11.3% of the variance. Okay. You see a huge drop from PC1 to what? PC4, right? So I'd imagine PC5, all this is not going to explain anymore. So probably going to include more than 3 PCs. From PC1 to PC2, you definitely need. From there on, you see it drops. Question?
Can you ever go backwards and say which SNPs are contributing most to the variation that you see?
For sure. Yeah, because so that's what PCA is. PCA, each component is just a weighted average of all the SNPs. So trust me, PC1 is telling us are the ones that are informative for having African or non-African ancestry. And you can actually do association study of PC on all your SNPs to find out which SNPs are driving the PCs. So you can easily do that. So that's how a quick and dirty way to do it, find out, okay, which SNPs are actually driving PC1. Just do regression on PC value onto all your SNP. Do a GWAS, but your outcome is what? PC, principal components.
So can you use that then as a scaffold to figure out your SNP, what kind of ancestry your SNP has? So you run principal components analysis. If you're
When you say what kind of ancestry is
well, like if you're looking at the admixture again, and let's say that you're interested in how many times your population has expressed. That's the word, but so I guess in order to find the flips where you have blocks of population stratification, would you use that in order to find those flips?
No, so you mean like what ancestry SNPs are? Yeah, not so much, right? Because these SNPs are just telling us that there are, in the ancestral population, there are large differences in the ancestral population. It's not telling us about necessarily the admixture where there's flipping with admixture, right? So those SNPs that are driving those, for example, a SNP that's monomorphic, let's say it's 100% in Europeans and 0% in African, that's totally going to be driving PC1. But that doesn't tell me where a flip is, right? Where, I mean, you have to do more sophisticated approaches, like I was talking about, like actually look at the genome combined and see like where the changes occur, and model like a hidden Markov model. It's a little more complicated than that. So this, but this is just basically telling us, if you look at which SNPs are driving the PCs, those are the SNPs that are informative for ancestry, okay? But not as informative as far as like, where the breaks are, where recombination occurs. Right? Because this tells us more about the ancestral population. Okay. We could also, someone asked about pruning. Do we need all 150,000 SNPs? So SNP relate has a nice function to prune. So now I'm doing prune the genome file and only give me SNPs that are no more correlated than 0.2, that have an R squared of 0.2 or less. So I'm pruning it. So SNPs that are highly correlated are going to be excluded from the analysis. So now I'm going to do the same analysis, PCA, but on a smaller subset of SNPs. So if you see, the pruning set now has 41,000. So we went from 150,000 to 41,000 SNPs. Okay, so let's see if we get structure if we only use a third of the SNPs. Okay. Okay. So here it is. If we just use a third of the SNPs, so you get this, essentially the same structure. A little bit snipped out. Okay. So you still are able to capture the structure with a small subset. Okay, so with that, if there aren't any questions, let's take a break and we'll see you at 3:30.